Hi everybody. In today's lesson, we are going to be doing a bit of a recap of things that you've probably covered in year 10 or year 9 science, but really introducing DNA so we can get into our next topic looking at genetics. So today we're just going to look at the structure and pairing of DNA molecules. We'll look a bit more at chromosomes and how they're actually structured using DNA. And then we'll look at these things called karyotypes towards the end. Like I said, really important information for the rest of outcome 2 in unit 2. So please make sure you're taking notes as you go through this, and you'll probably want to draw some diagrams in your notes as well. So as I said, this topic we're going to start looking at genetics. You'll notice I've written this in yellow. I'm going to use yellow for anything that relates to a field of scientific study. Okay, so genetics is a type of study. As you know, all living things inherit DNA from their parents. Okay, even if that is asexually, the parent is still that organism that gave rise to the offspring. But we're mostly going to be looking at sexually reproducing organisms when we look at genetics. But all living things have DNA. DNA is a genetic material that carries the information to synthesize proteins. And proteins, as we've talked about very briefly, are molecules that basically do everything in an organism. They determine structure, there should be a comma in there, they regulate growth, they operate all the different functions in an organism, and then they're involved in regulating things through things like hormones. So proteins are just really, really, really important molecules that are made based on the DNA code. Genetics, as I've said, is the study of the mechanisms and patterns of inheritance of DNA. We'll look at the inheritance a bit more as we go through this unit, but this is basically looking at how we end up with the DNA that we have. Like I said, we're going to focus today on the structure of DNA and chromosomes. It's really, really important that you understand this as we go through the rest of this unit. So DNA is made up of individual molecules that are called nucleotides. Okay, and if we looked here on this diagram, that is one nucleotide. Each nucleotide consists of 3 parts. Okay, it's got a deoxyribose sugar, which is a big name, but that's where DNA gets its name. So DNA is deoxyribonucleic acid. So D from the deoxyribo, N from the nucleate, and A from the acid. That's to do with this sugar. It's a deoxyribose sugar. There's also a phosphate group, which is shown in that circle there. And then each nucleotide has a nitrogen base. Okay, so you could put a B there for the base. But as you can see, DNA is made up of thousands of these nucleotides built together. Okay, so the way it bonds together is the phosphate of one nucleotide binds with the sugar of the next nucleotide. Okay, so they end up creating what is called this sugar phosphate backbone, which is, kind of like this spine that goes all the way down each of these strands of DNA. You should remember that DNA is a double-stranded molecule. So we've got one strand here, we've got another strand here with its sugar phosphate backbone holding those strands together, and then between the bases of each strand there are hydrogen bonds that hold those 2 strands together. And there's a particular term that we use here, that these strands run in anti-parallel. So parallel is when 2 things run side by side, Antiparallel means that these strands are built upwards. Sorry, these nucleotides build upwards, and these ones actually build down. Okay, so they're facing the opposite direction. That's what we mean by anti-parallel. And then here's the picture you're probably most familiar with, which is that famous double helix shape. So if we were to, have a much bigger molecule of this here, and then it would wind into that double helix kind of ladder shape, that's the form that DNA holds within the cells. Okay, so once you've written this down and drawn some diagrams, just jump into the quiz and have a go at questions 1 and 2. Okay, so DNA has 4 different possible bases. So remember, the bases are these here. Those 4 bases are adenine, which is denoted by the letter A, thymine, cytosine, and guanine. Okay, this is the same for every living thing. Okay, DNA only has those 4 bases, but obviously what makes different organisms different to each other is the combination of these letters, and that's stuff we're going to look at more later. And hopefully you remember this. The base pairing rule is that in DNA, adenine, or A, will only pair with thymine. So you can see that here, adenine and thymine pair together, they create these hydrogen bonds between them. And cytosine will only pair with guanine. And again, they will form hydrogen bonds together. Okay, adenine cannot pair with cytosine, guanine cannot pair with thymine. They can only pair with their complementary base pair. Okay, so that's a good term to put in there, complementary base pair. Okay, so just go in and answer question 3. All right, we're going to look a bit more now at how DNA is structured into chromosomes. So DNA, as we've just gone through, is the material. Okay, so this here is DNA. It's in that double helix shape, and it's made up of its individual nucleotides. A gene is one section of DNA, okay, and it's a section of DNA that provides the code for a specific protein. Okay, an example of this, so insulin is a hormone that regulates blood sugar levels. Insulin is a protein. Okay, so that's the product, the protein there. The insulin coding gene has the genetic instructions to make that insulin molecule. Okay, so obviously we have tens of thousands of genes for all of the different proteins that our body produces. DNA wraps around these proteins called histones. Okay, so each of these little blue circles is a histone, and the job of a histone is to allow DNA to wrap around it so it can be really, really tightly condensed. So if you think of a ball of wool, if you just tried to scrunch a ball of wool, it'll only scrunch up so much, but if you wound it really, really tightly, you can fit it into a much smaller space. Okay, so histones are the proteins that the DNA material wraps around. When we have the DNA and the histones combined, we call that material chromatin. Okay, so this whole thing here, which is the DNA and the histones, is called chromatin. Okay, I know it's very similar to chromatids, which we've looked at, but chromatin is the material, including the histones. And then a chromosome is the organized package of DNA. Okay, so this chromatin winds up into what is called a chromosome. And we're going to look at these a little bit more, but as you know, chromosomes are found in the nucleus of eukaryotic cells. It's different in prokaryotes. A chromosome is just loose within the cell, but we're mainly looking at eukaryotes here. And each chromosome contains specific genes. Again, we'll come back to that in a minute, but just do a little diagram of this. Obviously, you don't need to draw it in that much detail, but you could even just draw something like that being a chromosome, and then show that as it winds out, you've got your DNA, that is the material that makes up a chromosome. And then just have a go at questions 4 and 5. Right, looking more at chromosomes now. Different chromosomes carry different genes. And remember, a gene gives the information to make different proteins. Okay. What I mean by this, let's have a look at, say, for example, chromosome number 1. You get 1 from your mother and 1 from your father. These two chromosomes, they're both copies of chromosome 1. So they're going to carry the same genes. Here we've got chromosome 4. Chromosome 4 carries different genes to chromosome 1. So this is just an example. This is not actually what chromosome 1 holds information for. But let's say chromosome 1 has information for your eye color and your hair color, okay, as an example. And chromosome 4 carries the information for your height and your face shape. Okay. Again, these are examples, and they're often a lot more complex than that. They carry different information. The size of the chromosome is going to be different depending on how many genes it has, and then the length of each of those genes. Different genes have different numbers of bases within them. Diploid cells, as we've looked at quite a lot recently, have 2 sets of each chromosome. So we've got 1 from your biological mother, 1 from your biological father. So each of that is a pair of chromosomes, which we call our homologous pairs. Now remember I said these chromosomes carry the same genes, but they may have different versions of those genes. And we'll come back to this more in the next lesson. But if we come back up here, remember, let's say we have the eye color gene. You might get the gene for brown eyes from your mother and the gene for blue eyes from your father. So it's still the same gene, but different versions of that gene. Just jump in and have a go at question 6 once you've written that down. Okay, another term you need to know is karyotype. And this is basically just the term for the set of chromosomes within a cell. So like in that last picture, it showed all those chromosomes in the nucleus. We call this a karyotype. Now, as we know, humans have 23 pairs. 23 plus 23 in a diploid cell. The first 22 chromosomes, or 22 pairs, are called autosomes. Okay, that just refers to the fact that they carry the same genes. Okay, again, it might be different versions of those genes, but they carry the same genes. The way they are numbered in a karyotype, which is basically, a representation of those chromosomes, is from largest to smallest. So the first pair are the largest chromosomes because they carry the most genes. And then the 22nd pair are the smallest ones. They carry the least number of genes. And then, of course, we have the 23rd pair, which are the sex chromosomes. They are sometimes called heterosomes. And I'll explain that in a second. So we had autosomes and then heterosomes. The 23rd pair, if it consists of 2 X chromosomes, that codes for a biological female. And if it contains an X and a Y chromosome, it codes for a biological male. The reason they are called heterosomes is that if you look at for males, those 2 chromosomes are different to each other, X and Y, whereas the rest of them, they carry the same genes. X and Y chromosomes carry different genes. So these can be different to each other if it is a biological male with an X and a Y chromosome. So interestingly, obviously a female only has 2 X chromosomes. So when a female, if a female gets pregnant or it has an egg that is fertilized, it can only pass on an X chromosome to its child. A male has an X and a Y chromosome, they will get separated during meiosis. So a male can either pass on an X chromosome, so a sperm can either have an X chromosome in it, and that would produce a biological female, or the male could pass on a Y chromosome, which would result in a biological male child. So just using that information, have we got question 7? The very last thing we're going to look at today is just what can happen with abnormal karyotypes. So this occurs when a person is either missing a chromosome or has an extra chromosome. Now, there's certain chromosome pairs where if this happens, a zygote won't develop. It won't be viable to produce an embryo and then produce a baby. But there are several examples of where this can absolutely result in a fully functioning, healthy person. Examples of this are Down syndrome. So Down syndrome happens when there's an extra chromosome at the 21st pair. And this is actually officially called trisomy 21. So 3 chromosomes at the 21st pair. Another thing that can occur is results in Klinefelter syndrome, which is when a male who has an X and a Y chromosome has an additional X chromosome. Okay, so you see down here there's 2 X chromosomes and a Y chromosome. That person is still going to be biologically male because they have the Y chromosome. It carries the information for the male genetic organs and for the testosterone levels, but they also have an additional X chromosome. And so that's often called 47 because it's an extra chromosome, XXY. And this can result in, it's often not picked up until someone has gone through puberty, but often because of the additional levels of some of the female hormones, difficulty putting on muscle mass, they may carry a bit more fat, which is more common in female bodies, and it usually results in little to no sperm production, so that can result in infertility. Another version which occurs in females is Turner syndrome, when either an entire X chromosome is missing or it is partially missing. Okay, so obviously females should have 2 X chromosomes, but they can be missing half or all of a chromosome. Again, it results in some developmental issues and also infertility. The last question, I don't want to give you too many hints. I want you to have a think about it. It ties really well into what we studied in the last outcome. But just have a go at question 8, and I want you to be thinking about what is it that can occur that results in these abnormal karyotypes. And then that is it for today. Thank you.